Right, start up all. Okay, good afternoon, everybody. Right, sit down. And from this point onwards, we are shifting from grade 10 to the grade 8. So, successfully we completed grade 7 contents, and most of lessons we did as the online lessons. Right, here in this year also, we can experience the both. I mean, the online students, you can watch these videos in the same manner and participate the worksheet class, and I'll explain how we do the worksheet class in the physical class. Okay, right, this is the plan. Number one, for the physical class students, then our fixed date is Wednesday 2:30. That is the fixed date and time, 2:30 p.m. So this is our first lesson. Therefore, today we are not going to discuss any worksheet, right? But next week onwards, we will discuss the worksheet as well, right? The plan is this. In the first class, we will do the theory, and we can complete the notes and every stuff in the tute. That is the first thing. Then uh, next week, you will come. Next week, we don't have classes. I mean, the next means it is 27th or 28th. 29th, yeah, 29th we are not going to do the classes, right? Our next class is January 5th. 5th of January, you have to come with completed worksheet. Worksheets will be uploaded as usual, right, to the WhatsApp group, or once we make our website, we will upload it to the website. Okay, so you can take the or download the worksheet each and every Sunday, or probably Sunday, on or before Sunday. Definitely I will put it. That means the today we just record the recording. We will go with the recording, and for the online students, they can watch the video, and with that video I will upload the worksheet as well. Got it? You must do the worksheet as a homework, right? So for the physical class students, I mean to you, we will discuss it in the next class. After finishing the theory half at about 4 o'clock or 3:45, we can finish it. Thereafter we will take little time to discuss the worksheets. So as in the last year, you have to maintain separate book to write the answers for the worksheets. We did it very well in the last week, right, last year. And the next thing, for the online students, I am talking to the online students who are watching this in this as the video. Online students can watch the video once we uploaded it. You will get a message when we uploaded that one. So that is the first task. So watch the video after the before or not before the Sunday of that week. You can watch the video. Thereafter, their worksheet will be discussed. Then theory class will be completed. Then their worksheet will be discussed. Worksheet discussion will be held as the Zoom meeting. Physical class students, you also can participate the Zoom meeting if you are willing to do that. Okay. So worksheet discussion will be held to the online students on our usual day. I mean, this is the grade 8 class. For the grade 8 students, we did our normal Zoom meetings we held on Tuesdays, no? Yeah. So therefore, the Zoom meeting continuously continue in the Tuesday 5 o'clock. So worksheet discussion, the online students must definitely participate the Tuesday 5 o'clock to ask the questions if you have any doubt, and also to just engage in the kind of discussion. You have to participate the Zoom meeting at each and every Tuesday. And Zoom meetings also we will start January. That means the 5th or no, sorry, 4th of January. The first Zoom meeting will be held on 4th of January, right? If you can't come to the physical class on 5th, then you have to come to the Zoom meeting. Is it clear? So you can cover up the worksheet discussion. Later on, you can watch uh, the fifth, I mean the next video, I mean the next lesson, right? That's it. Is it clear? Right, that is our plan. So I think it is clear to the online students also. Then you are watching this video now, and after finishing or after completing the note and tunes and every stuff, then you have to come to the next Tuesday worksheet discussion on the next, I mean the Zoom meeting. Right. So our plan is clear then. This is, I took around 5 minutes to explain this because this is important. And now all the students who came here, you have collected the tute. You know this. You have this one with you. And the online students, many of you or your parents have visited here. Some students might have received that via post, and you have grade 8 first unit with you. Let's see what we have to do in this year, right? This is our plan for this year. These are the lessons that we need to complete, right? So you can see this in the screen. Look at the screen here. We need to complete 15 units, 5 for each term, 5 units for each term. Importance of microorganism is the first lesson. Animal classification, diversity and functions of plants, properties of matter, sound, magnets, electricity, changes in matter, human organ systems, electricity, plant processes, life cycles of living organisms, food preservation, solar system and natural disasters. Those are the 15 lessons that we need to complete within this year. So we will start the first lesson today, that is the importance of microorganisms. Wait a bit, I think something happened. Okay, we are talking about the importance of microorganisms, right? And I hope to do this one today. These are the topics that we hope to cover up in this video or this lesson, right? Microorganisms. Under that, we will learn what are microorganisms. Second, the appearance of microorganisms. And 1.2, effect of microorganisms. Right? Under that effect of microorganisms, we can't discuss all the contents here, but some of contents regarding the effects of microorganisms can be discussed. Okay, so you can take the tute and just see what you have, what is given in the tute, right? Okay, basically these are the main things that we hope to discuss in today's lesson. First question, what is meant by a microorganism? What is the meaning of microorganisms? I think you all have heard this term. This is not a new thing. In grade 6 we started a little part regarding the microorganisms. In grade 7 also we learned this word. We met that word in different contents. So as you think, what is a microorganism? Yes, yes, Dhinidhi. Okay. What she said is an organism which we can't see to our naked eye. No, that is the basic thing. An organism which we can't see to our naked eye. That's correct. Because this word is made of two parts. One is the micro, other part is organism. Right. The meaning of micro. Micro means very small. Right. Micro means very small. Not just small, very small. They are very small creatures. Organism means living being. You know this. In grade 6 we learned this word. Living things. Living things. So, simply the meaning of microorganism is very small living things. No, very small living things. As he said, we can't see them to the naked eye. How can we see them? Through a microscope. Last year we learned about the microscope. No, I think it is about 10th unit. We learned about microscope and how to use microscope. What are the types of microscopes that you know? Simple microscope, compound microscope, electron microscope, light microscope and electron microscope likewise, right? Through microscopes we can see them. And another big question, what type of microorganisms, what sort of names you have heard about the microorganisms? Amoeba, paramecium, yeast, amoeba said, yes, chlamydomonas, right? And what else? Fungi, alga or algae, okay. So different names we have heard. Viruses, correct, yeah. We can't figure the, yeah, then bacteria comes, right? Now you said number of things. Bacteria, viruses, protozoans. Have you heard protozoans? The word? No. Yes. Amoeba is an protozoan. Algae types, fungi types. Okay. So there are different types of micro. Basically, we can classify microorganisms into five groups, five main groups. Now let's check whether we can remember them. Right? As you said, I will write the first letters from the ones you said. Bacteria, then fungi, protozoans, algae types, and viruses. Right? They are the five different microorganism types. So, microorganisms are very tiny. And my last question, are they useful or harmful to us? Some are useful, some are harmful. Are there any microbes which are not useful or harmful? I mean the neutral ones. Not useful to us or either not useful nor harmful. Yeah? Any? Yeah? Plankton types. Maybe. There are many microorganisms which are not making any harm to us. Also which has no any particular use to us. Got it? Therefore, microorganisms, these type of microorganisms we can classify as useful ones, harmful ones, or neutral, which are not harmful or useful. Got it? Right. Then that is the basic introduction to the microorganisms. You can see a nice cover page here. Take the tute and see the cover page. Cover page, this one. Cover page. Yes. Right? Yeah, cover page means front page. So in that one you can see those categories: bacteria, fungi, algae, protozoa and viruses. Different types of them are given here. Today we can observe them and we can study more about different types of microorganisms. Right. The main plan of this lesson, mainly we will learn about the importance of microorganisms. How can we use the microorganisms usefully to our works? Okay. That's the main purpose. Right. Now we know what are microorganisms and we can continue the concept map. Right. Let's complete the concept map. Turn to the page number 1. It's given. Microorganisms are given. Now you can guess an answer to this. Microorganisms are seen through, can't see that. Yeah. Look at the screen. Microorganisms are seen through microscope. Okay. Write it. Complete that. Microscope. Next, microorganism types. 5 types. What are the types? Bacteria, fungi, protozoa, algae, virus. Okay, write that 5. Bacteria, fungi, protozoa, algae and virus. Bacteria, fungi, protozoa, algae, virus. Finished? Then microorganisms are very good. Living things. Living things. Microorganisms are living things. Microorganisms can be useful, harmful, or useful. Harmful or useful. Some are not harmful or not useful. Can be harmful or useful. Next. As they cause, why they are harmful? Harmful as they cause diseases. Some cause diseases. Some spoil food, spoilage of food, and some are molds. Especially molds are growing on the wet clothes, right? That black mold types. Okay, write them. Diseases, spoilage of food, and molds. Then the next. Useful in food industry. You can learn some little part regarding that. Useful in food industry. Not and useful for decomposition. For decomposition. Useful in food industry, useful for decomposition. Right. Then, what are microorganisms? We learned that. Let's write this. Before this, let's see this simple description. You can see different cells and molecules and atoms here with their sizes and scale. Okay. Here you can see these are the animal and plant cells. They are big actually. Human egg cells, pollen, frog egg, right? Some cells. Red blood cell in our body. Mitochondria is an organelle found in the body. And these are very tiny. Look at this. This is a bacteria, right? I will try to zoom this now. Look at the bacteria here. Bacteria. Smallpox virus, flu virus, polio virus. They are in the range of micrometer to nanometer range, right? Their size is smaller than micrometer, right? This symbol is used to micrometers. We know meters, centimeters, millimeters. No, micrometers are even smaller than that. Micrometers and nanometers, they are small. That is another reason to say that say them as the microorganisms. Micro size organisms. So look at this. These microorganisms are visible under light microscope. Is it clear? And they are visible under electron microscope because they are very tiny. Viruses are visible under electron microscope. Okay. And look at the different types or different photos of microorganisms. Some of them are given in the cover page also. We saw that, right? Cover page. And out of them you can see some organisms, or the bodies are made of single cells, only one cell, single celled organisms. We call such ones as unicellular organisms. Unicellular. Uni means one, right? Unicellular means one celled organism, single celled organisms. And some looks like multi-celled ones. They are multicellular organisms. Multicellular. So most of microorganisms are unicellulars. They are made of only one cell, right? Some of them are multicellular, right? If you see your tute, then you can see, right? Check out the tute, first page. If you see the tute, then you can see something like unicellular and microorganism. Left side is given, no? And introduction is given from top to down. All the microorganisms are living things or organisms. Likewise, microorganism may be single celled, unicellular. In the front page, first page. First page, left side. First page, left side. First page, not the front, not this page, cover page. Page number 1. Can you see left side? There is a description here. All the microorganisms are living things or organisms. Microorganisms may be single celled or unicellular, right? And also you can see the other sentence. Some microorganisms have more than one cell. They are multicellular. Can you see that one in the tute? Find it in the tute, right? Okay. Now let's write the note. What is the note? Here's the note. It's about the meaning of microorganism. Page number 1. Page number 1. The unicellular or multicellular organisms, the unicellular or multicellular organisms which are invisible, the unicellular or multicellular organisms which are invisible to the naked eye, to the naked eye, when taken individually. To the naked eye, when taken individually, are called microorganisms. Are called microorganisms. When taken individually are called microorganisms. Microorganisms. Nice. Finished? Okay. Then let's go to the next. How to observe the microorganisms, and also right, finish it first, hurry up. Unicellular, multicellular organisms which are invisible to the naked eye when taken individually, when taken along as one unit, are called microorganisms. Right. Then as we discussed, we can't see them to the naked eye. So we have to use a device. Maybe the light microscope, maybe the electron microscope. Right? That's what we can select. If the microorganisms are very small, we have to use electron microscope. But here we are not going to talk about the use in electron microscopes to see them. There are some microorganisms we can see under normal laboratory or lab condition. Right? In normal laboratory conditions, we can observe some microorganisms. So here we are going to observe them under the light microscope, because we can magnify the organisms up to around 1500 times using normal light microscope or normal compound light microscope, right? Therefore, there are some organisms we can observe under that I am not going to do that here in the class, because it takes time, right? If we start that here, we will take around the whole day to, I mean the whole time to observe them completely. Okay, therefore I will show you pre-recorded video of observing these three creatures. Look at the tute again, page number 1. How to observe microbes: bacteria, fungi, and protozoa. Okay, so in this video, when we are watching that one, I will show the method as well. You have to watch out for these shapes. If you want to notice the bacteria, just watch out this type of shapes. Okay, then you can identify bacteria, because I can't point out them while the video is, or while this one is playing. You can't, I can't point out this is a bacteria or that one is likewise. Okay, therefore just see, or the watch out for these shapes. Second, fungi. If you want to see the yeast fungi here, then you have to watch out for this shape. If you want to notice the protozoa, then watch out for this type of shape. Okay, shall we start it? Okay, this is the first one, right? The first task is observing the yeast. No, wait. This is fungi in the yogurt, right? This is what we do here. We get a slide here and apply yogurt culture over that. Using yogurt, we can see bacteria. There's a bacteria called lactobacillus bacteria. Okay, now we are going to observe the lactobacillus bacteria existing in the yogurt. Right, first thing, observation of bacteria. Applied some yogurt and covered this with the cover slip. We know how to fix the slips, because last year we learned that, no, under the microscope lesson. So this is what we can observe. Now you have to watch out for that shape. I said particular shape of bacteria. This is under 40 times magnification. You can see the magnification at one corner. Then we magnify to 100 times. Can you see kind of round circular some ones? Those are the bacteria. Zooming further, 400 times. These are the clusters of bacteria. Clusters, not the individual ones. Okay, groups of bacteria. So once you magnify to 1000 times, then you can see the bacteria. I have named that one. Can you see this? Bacteria is that tiny dot like thing is the bacteria. Can you notice them? Those are the bacteria. So they are visible under 1000 times magnification, right? So it is 1000 times magnification. Streptococcus bacteria is visible here. So watch out for that round shapes. Next, we can observe further and notice how bacteria are moving in this culture. 1000 times. Now it is clear, no? Very clear, right? Those are the bacteria types. Lactobacillus bulgaricus, lactobacillus bacteria living in yogurt. Actually, they are the ones which convert the milk to the yogurt. Lactobacillus bacteria. Right. This is how we can observe the bacteria under light microscope. Right. Normal light microscope can be used for this. But we are observing under the high power magnification. Right. And microliters of the yogurt culture is used, but we saw number of colonies. No, that clusters are the colonies of bacteria, right? thousands, or not thousands, millions of bacteria live in here. So are they harmful bacteria or useful ones? Useful, harmful, useful, no? We eat yogurt with that bacteria. They never harm us. Bacteria are the ones who make this yogurt. Actually, milk is converted to yogurt by them. Therefore, those bacteria types are useful ones. Second, then I will show you how to observe fungi types. Right. Here we are going to observe the yeast. Right. Look at this. Small amount of yeast has put into a kind of growth media. Especially we use sugar for this. Okay. Now around 20 minutes time is displaying within 3 or 4 seconds here. So this is the culture we take, sugar culture with yeast, right? Look at this yeast. Actually yeast producing a gas here, that is the carbon dioxide gas, kind of gas bubble create around it. This is the action of yeast, right? You can see the shape now. Can you see that shapes? I ask you to just watch out for that shapes. Those are the yeast. Yeast is a fungi under 1000 times magnification. Okay, we are watching under the 1000 times magnification. Right. That is how we observe fungi. Yeast. Next, a protozoan. Right. Let's see how to observe protozoan. In this one, in this culture, we can observe not only protozoan, some algae also. Look at the culture. I will show the culture to you. This is fermented water. Right. And let's observe how many creatures we can see here. Right. Fermented water culture is taken, and normally we can find that from a paddy field or old water pit or somewhere, this type of water, right? And we take small amount of that water into a glass slide, as you see here. We are taking a sample to a glass slide, then we can cover it with a cover slip, right, like this. So without covering, they are observing it here. So under microscope we can observe it, normal light microscope, right? So through that we can observe some creatures. Watch out for that particular shapes of paramecium that I asked you to observe, kind of shapes. Can you see that shaped organisms moving here and there? I asked you to observe particular shapes of protozoans. Now you can see those shapes. They are moving here and there, right? Many different forms of life are observed under that, right? Most of them are protozoans, and some diatoms. Diatoms means some algae types we can observe under this, right? Very clearly you can observe that protozoa, right? It is the paramecium, this moving creature, right? Now it's very clear, no? Under 400 times magnification. And also you can get an idea, when we observe bacteria under 400 times magnification, we didn't see that sort of clear image. Got it? So here, even under 1000 times magnification, we observed only dots. But here very clearly we can see their size, and they are actually bigger than bacteria, larger than bacteria. That's why we can take this sort of clear picture. This is called paramecium, right? And another type of protozoa here. Look at this. This is under 600 times magnification, right? So, and also an organism reproducing here, making own copy, making a copy like this. This is how they reproduce. Protozoa cell dividing and producing a new cell, right? Kind of rare capture. right? They are organisms. As the other organisms, they can produce the young ones. Okay, different sizes, different shapes, and different uses of them. Protozoans actually have no any advantage. They are not advantages, or there are no particular uses of them, right? Then our task is to nothing to write in the first page about these observations. I have given the whole note. Then let's go to the next. Appearance of some microbes. Let's identify these microorganisms, right? Appearance of them. Few microbes. Can you guess who they are? Yes, by looking at their shape, can't you guess them? What are they? They are the bacteria. So label that in your tute. Set of bacteria given in page number 2. Can you notice the bacteria? That cluster of them in the left top corner. Did you label it, Pan? No? Name it, name it. Name that as bacteria. Just before this, you can see this creature. It is an algae type, algae. You can see the same image there. Algae. Its name is Chlamydomonas. Chlamydomonas. Green color organism. Chlamydomonas. Next. What is this creature? This is amoeba. Yes, amoeba. Then what is this? Paramecium. We observed them through that video. Paramecium. And two fungi types are given. One fungi type. Guess this? Yeast. This is yeast. Correct. Yeast. And another one here. Right? Mushrooms are boom out of them. They are the mucor or mold. Mucor types. Mucor or molds. Growing on bread or some places. Right. Now you know the shapes. Let's see the real ones. Okay. I will show another video to you to see the real microbes. We start that with the bacteria, right? And we observed this video just before, and see others. This is Chlamydomonas. Chlamydomonas under 40 times magnification. Then about 100 times magnification. We can further magnify and see 400 times magnification and a higher magnification. Then magnifying further to 600 times. Look, they look like green algae. They can do photosynthesis. They can produce food. This is 1000 times magnification. Can you see that small flagella here, like two parts? And also they can move like this. An algae type, Chlamydomonas. Look at the flagella. I just highlighted that. This is under 1500 times magnification. That is the maximum magnification of a light microscope. At the highest magnification, it looks like this. Then the amoeba. It has no particular shape. When moving, they change their shape, right? by making a kind of particular feet like parts, they are moving like this by changing the body, gradually moving, right? They are moving gradually, moving further and forward. Amoeba that under 32 times high speed. So you can see how they move. Another protozoan, amoeba. Then they are paramecium, right? We labeled them. Now you can see the real shape of them. They have hair-like structures in their body. Clearly, they can locomote using that hair-like cilia. That's how they look like. Just like this. I think under very high magnification, you can see that hair-like stuffs. Is it clear to you? That hair-like structures. Okay. That's how they stay in their micro world. Then the mold or mucor. This is black mold grown over food. When you zoom, it look like that. Actually, they are filamentous. They look like small fiber, like tiny hyphae there, right? Filamentous creatures. This is a community. They live together. And when we zoom further, you can see that filaments or the hyphae of them. Black dots are visible. They are the reproductive structures. Then under 30 times magnification, just 30 times means the fungi types are bigger, no, large. Especially this fungi, mold, the mucor type is larger. Under 30 times magnification, we can clearly see them. This is 40 times magnification. Under 40 times magnification. So further magnified and 30 times. No, then it's clear, very clear. This is the appearance of mucor. We observed the yeast. Therefore, I'm not going to show you the yeast here. Right. That's really clear. So that collection of clips are about the different microbes, right? Collection of them. Right. Then let's go to the page number 2. And 1.2, effect of microorganisms in food. So we will learn about different effects of microorganisms, and you also said some effects, and in that concept map also we wrote microorganisms can be either useful or they can be harmful. Useful or harmful. So we can learn their uses or harms, right? The both are studied or discussed under one topic. We learn them as effects. So there are useful effects of microorganisms and harmful effects. Let's put the harmful effects, or to discuss the harmful effects, the next week, right? In the next lesson we can learn about the harmful effects. Today we can learn some of their useful effects, some of, right, not all the ones, some of the useful effects. So we are talking about how we use microorganisms useful in food industry, right? What sort of food are made using microorganisms? Yeah, bread, correct? Buns, yogurt, curd, cheese, butter, really? Okay. Yogurt, curd, cheese, correct? Bread and buns, correct? What else? Some biscuit types, maybe. Biscuits, probably in bakery industry, we can put it as commonly as bakery industry. Dairy industry, dairy products means the milk related products. No? And also we use it in vinegar production. Vinegar. Some beverage industries, right, like wine industry, they are useful. Microorganisms are useful. And here we will talk about two applications of microorganisms. First one, the bacterial support, other one, the fungal support. So first one is how to make the curd, right? First topic is that, wait a bit, making the curd using oil and other stuffs. I just want to find a small video for this. Let's see this. No. I don't have a video of making the curd production. I will search for one. Right, this is it. How to set curd or yogurt? We need buffalo milk, and when producing the curd, 1 liter of buffalo milk, yogurt as the culture, right? This is how a professional doing the work. So let's see the steps. First thing is heating that to around, first we boil it, right? First we boil that, thereafter putting that to the pots, maybe the clay pots, right, or maybe plastic cups we can use for this. And then we will fast forward this, that boiled ones were poured into that. Then this is the next step, right? Most important step is this: we have to keep it until it cool down to 40 degrees Celsius, around our body temperature. Then after we need to introduce this culture medium. This is very important. Introducing the pre-made curd. That means we are introducing the bacteria into this. Is it clear? Right? First we boiled the milk to destroy the present microorganisms. Boiled that. Thereafter let it cool until it comes to around 40 degrees Celsius. Because at that temperature they are growing very fast, microorganisms, especially bacteria. Then introducing the sample. That is the next important thing. Sample was introduced because in curd, naturally lactobacillus bacteria does not exist, right? Therefore we add or they introduce a sample, not the whole pot of curd. We took only a spoon, no? That is enough, because once we put that to the curd pot or the milk pot, the bacteria in this sample then start their work, right? It will take around 12 hours to complete the whole process, right? Then after mixing the sample like this, after mixing the sample, we will let, we will keep it until it settle. Around 12 hours we have to keep this, right? So you should not keep that in refrigerator, okay? Just outside in a certain cold place we have to keep it. If you keep it in the in a refrigerator, the process will not take place because bacteria can't act on, no? Therefore always we have to keep it here. Then this is the process which occur. Milk particles and flat globules are found in the milk. So that cluster of them, they clusterize, and look at this. The introduced lactobacillus make small clusters here. Then what happens? The liquid curd converts to solid material like this. By gathering them, kind of semi-solid substance form. That is the curd. So only bacteria can do it. We don't have machines to do this process. Is it clear? Then when we make in curd, we can't use a machine for this. We need to take the support of bacteria for this. Only bacteria can do the process. So this is the process of making curd or yogurt. Same manner we make the yogurt, but the ingredients, we add some other ingredients to the yogurt to get that different taste and different setting process. Okay, right. Look at the tute. How can we change milk into curd at home? Write this here. Wait a bit. I just want to go to the next slide. The bacterium lactobacillus, the bacterium lactobacillus, the bacterium lactobacillus promotes the formation of curd. The bacterium lactobacillus promotes the formation of curd. Promotes the formation of curd. Full stop. It multiplies in milk. It multiplies in milk and converts it into curd. And converts it into curd. Converts it into curd. Converts it into curd. Finished? Okay, others also finish it quick. Then, for the next task, we can do an activity. I have prepared some stuffs to do this activity. Okay, let's do the activity. I think you can see that through the screen as well. So in this activity, if you see the tube, you can see the ingredients needed for this. Sugar, yeast, a balloon, warm water around 40 degrees Celsius, a bottle and a beaker. Right. I have prepared a bottle here. This is a round bottom flask. And we need a beaker. Let's take a beaker as well. Let's take a beaker. So here there is a beaker. You can't see that. I will just move the camera down. Right. We have a beaker then. And what else we need? Sugar, let's take sugar. This is sugar, right? Let's keep that also. And yeast. Here I have newly borrowed yeast sample because we can't use old yeast for this. Therefore, I brought it newly. Then let's prepare some yeast also. I will take that to a small petri dish or something. Right. Here this is a petri dish. I will put a sample of yeast to that. Right. This is how the yeast sample look like. Right. Through the camera you can clearly see this. So let's put a sample here to that. Yeast is ready. Then lukewarm water. I will take warm water to a beaker, another beaker. I think this water is too much hot. Let's measure the temperature. We need around 40 degree 40 degrees Celsius warm water. Let's check out the temperature of this. Seems too much. We'll see. So it is around 47 degrees Celsius. No? 51, 52. Still changing. I will add cold water, then I can add this warm water to it to make it around 40 degrees Celsius. Around 40 degrees Celsius. I think that is enough. No? We'll see the temperature. Yeah. 38.8. That temperature is suitable. Why do we add warm water here? Because the microorganisms grow faster in this temperature. Okay. If you want to increase the temperature a bit, I can add some more warm water. Okay, enough. Right. Then our task is to add these stuffs. You can see these steps in the tube. Right, page number 2. You can see the steps. Dissolve 2 teaspoons of sugar in 200 milliliters water. Around 200 milliliters. You can see that I have taken around 200 milliliters of water. And how many teaspoons? 2 teaspoons, not the tablespoons. Therefore, I have to add 4 tablespoons, right? No, 2 teaspoons. Teaspoon. That's it. Okay. No, this is teaspoon. So 2 teaspoons of sugar will be dissolved in this. Mixing up. It's dissolving. As this is warm water, actually it's dissolved a little bit faster. And about 1 teaspoon of yeast should be added to that. Okay, you can see the steps in the tube. About 1 teaspoon. Still it is not dissolved completely. We must dissolve that completely. Enough. Then I need to add around 1 teaspoon. Right. I think this is not full teaspoon. I will add some more. Like this. About 1 teaspoon of yeast. Then dissolve it again. Dissolving that again. Remember, this is not very hot water or not too much cold. Okay, so around 40 degrees Celsius water. Mix in this. Then we need to pour it to our bottle, right? Let's bring the camera a little bit close to the bottle. I'm going to pour that to the bottle. Here's the bottle, and I am pouring it. Pouring our mixture to the bottle. Oh. Right. Then we need to fix a balloon to its mouth. Fixing a balloon. I have a balloon here, and this is the balloon. Let's fix that to its mouth. Just like this. So balloon is fixed. Then what we want to do? We have to wait and see. Wait and see what happens here. Right. I think you can notice some gas bubbles, so air bubbles here in this place. Can you see? Few gas bubbles evolved here. And to take the result, it will take a while. At once you can't see the result. So now we can see slightly it is blown. Can you see that? Slightly blown. Just wait. We have to wait around 10-20 minutes. Okay. Right. I will show you the result time to time. Then till that we can discuss the process here. I think you can remember I showed you a video where the sugar and yeast mixing and observing it under the microscope. Uh, let's watch that video back. Let's find that video. This is the video. So under the microscope, if you observe the same solution, it will look like this. Right? Gas bubbles appear. So when we use a microscope to observe this, then that will look like this. Look at this. A gas bubble growing here. This gas is carbon dioxide. As yeast start the respiration, it releases the carbon dioxide. Okay, that's why a gas bubble or air bubble create here. What we expect to do in this activity also the same. Right? We just hope to notice whether that gas is produced by that. It takes time. Still we can't see the kind of particular result. But gradually when the process going on, when this yeast feed on the sugar, right? We added sugar and yeast. When it feed on sugar, then we can see the result. In bakery industry, we use it very usefully, right? First, I will show you a video. Thereafter, I will explain how do we use that in the bakery industry. Okay, right. Here's the one. Using this in the bakery industry or bread making process. Right, process of making bread, not this. Wait, not that is not the video. Wait a moment till I find the correct one. This is the one. See the ingredients needed for this. You can't see there. Here it is. Yes. yeast, water, oil, sugar, flour, and salt. Those are the ones we need. Then here's the process. Put in the yeast, dissolving yeast in glucose solution, or we can use uh, sugar for this. And bread looks porous, right? Porous means there are holes between that. To make it, actually we use this process. Put in water, put in that yeast mixture to that, or some oil to the yeast mixture. Thereafter we can add the wheat flour, finally little amount of salt, further add in more wheat flour, 2-3 spoons, and we make dough. This is called dough, right? And by adding some more wheat flour we can keep it. Then when it is keeping, it rise up. We call it is rising. Bread is rising. Can you see this? Rise up. It's puff up. So this is the process take place inside once we keep it for long time. Then we can make some balls, and just like this we can make the buns. So buns looks like that. They are not much beautiful. It looks ugly, but that is the final output. Wait. Again, wait. This just skip into the next. Right. So this is the output. That's the output. And they look porous because of that. And before moving to the explanation, I will move to the observation now. Observation of our activity. You can see that here. Can you see the balloon has blown a bit than the previous occasion? But still it's growing. Now I will explain how this carbon dioxide makes the bread porous, but let's see the observation. Now we are merely taking kind of observation here. It's very close, right? Can shake that a bit. So if a gas is producing here, that gas will fill into the balloon, right? As the yeast working on sugar, we can see this result. Oh, it is rising up, right? It took around 5 minutes to give this result, no? Yeah, just wait and see. That's very clear. It's slightly blowing now, blowing up, gradually shaking and rising. Right. It's rising along. Yes, it was risen. Right? It will further blown up. I mean, when the result is going on, that will look bigger and bigger. Right? We should give time for this. So how can we use this usefully? I mean, how do we use this process in bread production? We saw how to make the dough and make this bread, right? This is how we can do it. This is how it works actually. When we make the dough, small carbon dioxide bubbles will create, right? Carbon dioxide bubbles will be created by the yeast, right? The gas bubbles. So once we put it in the oven Once we put that in the oven and supplied the heat, the gas bubbles expand. They get bigger due to the temperature. When the bread is baking, so bread or bun or anything, when it is baking, it becomes further big. Also cake or any other thing that we make with the yeast, right? Rise up in the same manner. So it looks big because of this. So our product will be porous, right? The best example to understand that is the bun, tea buns, normal tea bun. If you take a tea bun and squeeze it, then you can find out the real size of the dough. Have you done that one ever? Just try it once. It's really small, right? Actually, there is very small amount of wheat flour in it, but the buns look bigger, right? Because of the yeast. That bakers must respect the yeast. If not, they can't give that sort of large product to us. Right? They are serving us a large product because of yeast. Yeast make it rise. So that's it. Therefore, we also have to taste it without squeezing, because we can't enjoy, we can't take the same, I mean the feeling. If you eat a squeezed bun, you will eat it for one or two gulps. Okay, that's the thing. Therefore, this process, yeast and sugar, action of yeast and sugar is usefully used. Again, let's go to the activity desk. Look at the size of balloon now, right? It's getting bigger. Okay, now let's write the note. Go to the tute. Page number 2, still we are in page number 2. The action of yeast, write this. Yeast reproduces rapidly. Yeast reproduces rapidly and produces carbon dioxide. Yeast reproduces rapidly and produces carbon dioxide during respiration. Yeast reproduces rapidly and produces carbon dioxide during respiration. Full stop. During respiration. Full stop. Bubbles of the gas filled in the dough. Bubbles of the gas bubbles of the gas fill in the dough and increase its volume. Fill in the dough and increase its volume. Increase its volume. Right. Again, we can give a glance to the activity. So it has grown further, right? Becomes a little bit bigger. Then let's see our observation. Here's the observation. Page number 3, not 3, still in page number 2, no? This activity also in the page number 2. Here there are few stages of the activity. Step 1, 2, 3. Third one, it looks very big. Write the observation. Inside that box you can write the observation. The mixture produces bubbles. The mixture produces bubbles. The mixture produces bubbles and balloon gets blown. The mixture produces bubbles and balloon gets blown. The mixture produces bubbles and balloon gets blown. Balloon gets blown. The mixture produces bubbles and balloon gets blown. Right. This is the final glance. So this is what we can observe really. This is the observation. Right. Remember, yeast and sugar can make this change. So that is useful. And yeast is a microorganism. What type of microorganism is the yeast? It's a fungi type, no? Fungi. Yeah. Right. I hope then this is what we plan to finish. Next week we will cover up or we will learn about the harmful effects of microorganisms and some more details about microorganisms. And for the online students, let's say goodbye and goodbye everybody. So this is the end of the lesson.